Good day, everyone. We are the Group 13 and we're thrilled to present to you our topic, which is the genetic engineering. I am Ace Larigo and I am joined with my group mates Roy Lamba and Joshua Lucky. So when we say genetic engineering, it is a captivating field nga involved sa manipulating sa DNA sa organisms para ma-improve ang yang daan nga trait o ma-address ang mga genetic diseases. So it has the potential to revolutionize various industries including medicine, agriculture, and environmental conservation. So ang goal sa genetic engineering is to create organisms with the desired traits that may not occur naturally or to enhance certain characteristics. For example, ang scientist kay maka-genetically engineer sila og mga crops to be more resistant sa mga pests or diseases. O para maka-create sila genetically modified organisms or the GMOs that kanang maka-produce og higher yields or maka-develop sila bacteria nga maka-produce og useful substances like medicines. So, in our presentation, we'll delve deeper into the ethical considerations surrounding genetic engineering, its potential benefits, and the impact it may have on the society. The learning objectives. At the end of this chapter, the student should be able to first is to describe the principles in genetic engineering or the recombinant DNA technology. Second is to identify the environmental health and social implications of genetically modified organism or the GMOs and their potential future impacts. And lastly, to examine how GMOs are regulated. Genetic engineering. A genetic engineering, also known as a recombinant DNA technology. It refers to all techniques that allow desirable genetic traits from a plant, animal, or microorganism to be incorporated into an unrelated species. Think of it like a genetic mix and match game. Ang scientist maka-identify siya og specific nga trait nga ilang gusto, such as disease resistance or improve growth in one organism. And then ilang i-transfer kana nga trait into a different organism that doesn't naturally have it. For example, let's say na isa ka plant nga naa siya natural resistant to a common pest. So ang scientist makagamit siya og genetic engineering to take the gene responsible for that resistance from the plant and then iyang i-insert into a different plant species nga susceptible sa common nga pest. So ang bagong plant matawag na nato siya na modified na siya. And then we'll also have the resistant trait and can better withstand the pest. So, katong akong gi-example kaganina is parehas ra siya concept sa animals o sa microorganisms. Ang scientist maka-transfer siya desirable traits from one species to another. And then, it's important to know that genetic engineering is a complex process and scientists follow strict regulations to ensure the safety and ethical use of genetically modified organisms or the GMOs. So, in simple terms, ang genetic engineering is usa siya ka technique nga ga-allow sa scientist nga magkuha og desirable traits from one organism to another and then it transfer into a different species, creating new organism with improved characteristics. A genetically modified organism or the GMO is pares ra siya sa plant or animal that's been given a makeover inside. Ang scientist nag-change ang DNA which is like instruction manual to give it new abilities. This could be anything from making a fruit bigger or helping a plant fight off bugs better. So as you can see in this picture, here's a step-by-step process involved in genetic engineering. First is the DNA extraction. So scientists start taking out the DNA nga gikan sa nucleo sa isa ka cell, and then ang DNA is like a set of instructions nga nagsulti nga unsaon pag function sa isa ka cell. Second is the plasmid DNA extraction. Ang ga-remove pud siya small circular piece of DNA nga gitawag siya plasmid gikan sa bacterium. So, plasmids are like extra instruction manuals that bacteria can share with each other. Third is the gene cutting. Next, kay ang scientist mag-cut out of specific gene nga gikan sa DNA, and then kani nga gene siya ang magdala sa instruction nga gikan sa particular trait or characteristic. So, fourth is the plasmid cutting. They also cut and open the plasmid DNA to create a gap. So, the last thing is the DNA joining. So, they cut ends of the gene and the plasmid DNA are then joined together. It's like putting puzzle pieces together. So, kani nga DNA, maghimo siya bag-o nga loop of DNA o gitawag siya recombinant DNA. So, the gene becomes a part of the plasmid. So, by following these steps, scientists can create a modified DNA molecule that contains the desired gene. So, this modified DNA can be inserted into another organisms like plants or animals to give them new traits or abilities. It's like adding a new chapter to the instruction manual of life. So, this is the application of genetic engineering. One of the most common application is in the production of human insulin, which is a life-saving drugs para sa mga tao nga naay diabetes. So here's how it works. First is ang ginatawag nato nga plasmid DNA. Isa siya ka proseso nga nagsugod sa isa ka circular piece nga ginatawag nato plasmid nga makita siya sa bacteria. Plasmids are ideal for genetic engineering because ga-replicate niya independently ang bacterial cell nga ga-allow nga mag-multiply and maghimo og daghan kaayo nga mga inserted gene. Second is the DNA is cut with restriction enzymes. Ang scientist gagamit og special proteins nga ginatawag nga restriction enzymes para makat ang plasmid sa DNA of a specific sites. So this creates an opening where a desired gene such as the human insulin gene nga ma-insert siya. Third is the plasmid is reintroduced into bacterium. Once the insulin gene has been inserted into the plasmid, the modified plasmid is reintroduced into a bacterium. This is often done using a process called transformation, where the bacteria are made to take up the plasmid from their surroundings. Fourth is the engineered bacteria multiply producing insulin. Once inside the bacterium, ang plasmid na ang magdala sa insulin dito sa gene can replicate independently. As the bacteria multiply, they produce the human insulin protein. Last is the insulin is separated and purified to produce human insulin. The insulin produced by the bacteria is harvested, purified, and packaged into a form that can be used by people with diabetes. So, this process has revolutionized the treatment sa diabetes, making it possible to produce large quantities of human insulin in a cost-effective way. Genetic engineering has proven to be useful in many different fields or aspects of society. First is in the medicine, second is in the field of food industry, third is in the field of agriculture. Useful ang genetic engineering sa field sa medicine. Think about a genetic engineering like a tiny factory inside a bacteria nga makahimo og medicine for us. For example, we can put a small piece of human DNA that has the instructions for making insulin into bacteria. O gani nga bacteria, ay gagamit tag instruction para makahimo tag insulin, which we can collect and give to people who need it. So, second is the food industry. Ang genetic engineering kay more siya gahatag og superpowers sa atong food kay we can make changes to the DNA of plants to help them grow better, resist diseases, or even have extra nutrients. For example, ang scientist gahimog laing klase sa rice nga naa siya extra vitamin A. Ang gitawag ni siya o golden rice. Makatabang kaayo ni sa mga taong nagkulang sa vitamin. So, ninana ka-importante ang genetic engineering sa field sa food industry. Third is ang agriculture. So, sa farming, ang genetic engineering is like giving plants their own built-in bug spray or more bitaw siya sunscreen. We can change the plant's DNA so kanang maprotektahan nila ang ilang mga kaugalingon sa piste or sa lain nga panahon, which means we can grow more food with a fewer resources. So inana kay useful ang genetic engineering sa field sa medicine, food industry o sa farming. So these are the examples of economically important biological products.
The process of making insulin through genetic engineering, biological substances that have practical commercial applications that were once rare or scarce cannot be made readily available through genetic engineering. Other substances that were once available in small quantities through animals or plants are now available in large quantities through manipulations and alterations of genes. The genes of human insulin inserted into the gap in the plasmid is now genetically modified. The genetically modified plasmids introduce a new bacteria of cells. These cells divides rapidly and starts making insulin. Genetic engineering, also called genetic modifications, is a process that uses laboratory-based technologies to alter the DNA makeup of an organism. There are two samples of genetic engineering: a human insulin, human growth, and hormones. Adoption of genetically modified crops in the world. And there are two types of eggplant. The other side is non-BT eggplant and BT eggplant. The difference between BT and eggplant is that BT eggplant has the same nutrients compositions that has the conventional eggplant, except for the traditional BT eggplants. Proteins renders to the resistance to the fruit and the shorter borer. When introduced into plants by BT genes produce proteins, but siya ang produce of proteins that are toxic but harmless to humans and animals. The following are some successful genetically modified crops. Number 1, corn, soybeans, canola, sorbet or squash or potato that are square favor applications of herbicides or pesticides. Number 2, tomatoes that have a delayed ripening process of resulting in a more flavor or colors of flavor sour. Number 3, soybeans that are lower in saturated fats or offer better frying stability. Number 4, papayas that are resistant of ring spots virus. Number 5, peanuts with a high oleic of content. Number 6, golden rice with high vitamin E or beta carotene or content. Number 7, and apples that do not turn brown even after having been peeled in. And the last is number 8 is carnations that produce blue milky flowers. Issues and concerns. Genetic engineering is a controversial issue because of causes of concerns. Critics have branded GMO products as a Frankenfood based on a Mary Shelley's Frankenstein, a story about the scientist and ethical Victor Frankenstein who created a monster that turned against his own creator. This branch of criticism warns of the possible negative side effects of GMOs. Environmental concerns. The risk of crossings were in genes from GMO foods into wild plants and other crops and negative impact in insects and other species. Reductions of the other plants types of leading of the loss of the biodiversity. The following are common issues and concerns of GMOs related to health and environment. Number 1 is possible effects on non-target organisms such as beneficial insects, mammals, and wildfire, and engage the growth of threatened species in the environment. Number 2, possible genes flew to our crossings with the other varieties, including indigenous crops and with wildfire relatives. Number 3, possible formations of resistance insects, pests, and microorganisms. Number 4, possible persistence of toxins in the environment. And number 5 and last is possible formations of human diseases such as allergies and cancers. GMO contaminations. There is a possibility that modified genes that cross with the indigenous crops or wild organisms, for example, with the wide populations of BT corn, will sooner or later cross the pollinate native corn varieties or yellow wat corn that locally used as a food and animal foders. The contaminations of indigenous crops with genetically engineered DNA is a permanent will be extremely difficult, expensive, and eradicate, which can possibly drive indigenous species or crops of extensions. White corn kernels are characterized by theirs pale or luspasia nga pagkayelo, or corn is a vibrant of golden hoop.
Good morning, ma'am. Good morning, everyone. I am Joshua Tilaki, and my assigned topic is all about GMO risk. GMO risk. Scientists who are critical of GMOs are questioning the health and environmental risks that GMOs pose. Studies have shown that consuming and producing GMOs can result in a new and or more virulent strains of infectious agents. The prevalence of toxin in the soil, killing of non-target organisms, and other unpredictable and unknown long-term effects are also concern of GMO critics. The potential health risks to humans include the possibility of exposure to new allergen and genetically modified food, as well as the transfer of antibiotic-resistant genes to gut flora. The following are some examples of missteps concerning GMOs. First, Bt toxin and monarch butterfly. Bt means Bacillus thuringiensis. These toxins have been successfully used as a biosyntheside against caterpillar, beetles, and flies, including mosquitoes and black flies. Bt also synthesizes insecticidal proteins during the vegetative growth phase. which are subsequently secreted into the growth medium. Second is bovine growth hormones or POSELAC. POSELAC is a recombinant pharmaceutical used to increase milk production in lactating dairy cows. Second is bovine growth hormones or POSELAC. Human abnormalities have been attributed to drinking milk from cows injected with RBGH. The milk contains high levels of a protein fraction called insulin-like growth factor, which has been linked to the formation of breast, prostate, and colon cancer. And the third one is the terminal or suicide seed. Terminal or suicide seed. American agrochemical and agricultural biotechnological company, Monsanto, patented a technology that creates sterile seed to protect its patented synthetic genes called Terminator technology. By the media, sterile seed technology is a type of gene use restriction technology in which seed produced by a crop will not grow. Crop will not produce viable offspring seed or will produce viable seed with specific genes switched off. The inevitable offspring seed force farmer to buy. Issue. The issue of authorization is also present, particularly regarding who is authorized to use modified or altered genes. For example, the Bt toxin gene is protected by the patent of the agrochemical company, Monsanto. If Bt corn contaminates native corn varieties, Monsanto may sue local farmers or unauthorized possession of their protected gene. A patent is the granting of a property right by a sovereign authority to an inventor. So, ang GMO is mo connect po sa patent issue. Kung kinsa tong sa issue sa authorization of the variety. Ethical issue. Many critics claim that the technology of genetic engineering goes against religious belief because its essence revolves around the idea of playing God, as GMOs directly modify the flower function and processes of organism. However, major religious groups disagree on this genetic engineering. So daghan nga critics nga claim nga ang technology of genetic engineering kay ga-against siya sa religious belief because of the idea of playing God. Pero ang mga major nga mga dagko nga mga religious group kay ga-agree sila ni because mga plants and animals man nilang modify, not human modification. Genetic engineering in the Philippines. Agricultural biotechnology is recognized by the Philippines government as an effective tool in attaining food security, sustainable agriculture, and in promotion of health and food environmental safety. So, ang agricultural biotechnology na recognized sa Philippines because it helps them to food security, sustain nilang agriculture, and promotion of health and food environmental safety. Research involving transgenic crops is conducted at the following institutions. First is the International Rice Research Institute or IRI sa Los Baños, Laguna, which develop varieties of rice and works environmental sustainability in rice farming. So kani International Rice Research is mo ni siyang biggest nga mo rice research institution in the world and nag-help yun siya para ma-develop yun ang mga varieties ng mga rice. Second is National Institute of Molecular Biology and Biotech sa Los Baños, Laguna, which research on and develop programs to achieve sustainability to create better food and feed products, as well as medical drugs biotechnology. It aims to promote and implement research focused on prevention, early diagnosis, and treatment of infectious diseases that appear as major threats and primary causes of mortality in Philippines. So, kung siya ka-focus niya siya para mga early diagnosis and treatment para makatambalan to mga infectious nga mga diseases that major sa mga sa Philippines. The fourth one is the Institute of Plant Breeding, University of the Philippines, Los Baños, Laguna that handles the development and improvement of plants and crops other than rice. Ang Institute of Plants Breeding is yung mag mix sila duha ka plant and makabuo sila mas tiyada nga variety or new plants nga mas makatabang sa paglambo sa atong agriculture. And last is Philippine Carabao Center or Science City of Muñoz, Nueva Ecija, which is also attached to the DA and aims to conserve the country's Carabao population. Ang Philippine Carabao Center is nakafocus siya sa mga Carabao, how they handle it, and yun sa nila pagpadaghan. It also mandated to conserve, propagate, and promote the Carabao as a source of milk, meat, draft power, and hide to benefit the rural farmers.